A very good morning to you, and you're welcome to this morning's webinar. I hope you're keeping safe and well wherever you're joining us from today. My name is Mark Gibson, and I'm the manager of the Teagasc Connected program. This series is brought to you by Teagasc in collaboration with Dairy Sustainability Ireland, Food Drink Ireland SkillNet, and the National Rural Network. In Ireland today, agriculture, as we know, is responsible for approximately 32% of all greenhouse gas emissions. Methane is the main driver of these emissions, with almost 2/3 of greenhouse gases from agriculture arising from methane. And today, I'm delighted to be joined by Professor Sinead Waters, who is Principal Research Officer with Teagasc, based in the Animal Science Bioscience Department in Teagasc Grange, who will discuss today the development of strategies to reduce methane emissions from agriculture. Sinead, good morning to you. How are you?
Good. Thanks, Mark. Thanks for having me and the opportunity to speak today.
You're very welcome. Sinead, you're joining us from Galway. You're not, you're a stone's throw from me today. You're down by the coast. Sinead, can you tell us a little bit about the work that you're doing in Teagasc?
Sure. So as you said, Mark, I'm a Principal Research Officer in Teagasc in the Animal Bioscience Department. So the focus of my research is essentially on the microbes that exist in the rumen or the belly of the animal and its role in enhancing feed digestion, feed efficiency, and also, importantly, in reducing greenhouse gas emissions from agriculture. And that's the path really that I got into methane emissions research. So at the moment, I am working on a lot of projects funded by the Department of Agriculture, Food and the Marines here in Ireland, funded by Teagasc, funded also by the EU, on trying to understand the role of the microbes in producing methane emissions, but also in using that information to try and develop methane abatement strategies or strategies to reduce methane emissions from agriculture. And this could be, we're involved in work with the Irish Cattle Breeding Federation, which I'm going to speak about today, also with work on developing options for feed additives to reduce methane emissions from beef, dairy, and sheep. And also we're planning on some work on looking at multi-species swards as another option. So that's really what we're working on. So I come from a molecular biology background, and that's the route where I got into working in methane emissions research.
Brilliant. Really looking forward to your presentation. So,
Great.
Sinead, I think we'll go straight to the presentation, and if you could share your screen with us.
Sure.
And to remind everybody, if you have a question for Sinead, or if you'd like to put through a comment, please do use the Q&A tab at the bottom of your screen, so we can see your screen perfectly there now, Sinead. So we'll hand over to you, and we'll talk to you afterwards.
Thanks, Mark. So just in relation to methane emissions, more recently, methane emissions comes with very negative connotations. So today I wanted to speak about some of the strategies that we are working on in potentially trying to reduce methane emissions from agriculture and the research that's going on in Teagasc and elsewhere that can hopefully help us to reduce methane emissions. So just by way of an introduction, we're facing a major challenge in agriculture and that we're feeding a rapidly increasing global population projected to rise about 10 billion by the year 2050. However, at the same time, we're under international pressure to reduce the environmental footprint of this agriculture. And we hear about it all the time in the media. So, what is methane emissions? So, the sources of methane emissions are from ruminants, from the fermentation or digestion of feed, from management of organic waste and manures, and also from in Asia from the rice paddy cultivation. And that's how it's defined in terms of by the IPCC. So when I say biogenic sources, I mean the biological sources of methane. Agriculture is responsible for about nearly 33% or a third of Ireland's GHG emissions, as Mark said earlier. And methane accounts for the majority of greenhouse gas emissions from agriculture, therefore. The sources in Irish agriculture include enteric fermentation, or the breakdown of feed in the rumen or the belly of the animal, and also another roughly 10% comes from stored slurries and manures. However, reducing methane emissions is going to be key to meeting our EU targets on climate change. And you would have heard from Dale Cramen in the Department of Agriculture in recent webinars describing the pressure we're under to reduce methane emissions. As you'll see here from this graph from the EPA, the Environmental Protection Agency, the blue part of the chart here refers to the enteric fermentation. So this is basically the sources, the total emissions of agriculture, and enteric fermentation accounts for a very, very large proportion of that in the blue portion of the bars here. So methane is the second most important greenhouse gas that's implicated in global warming after CO2. And it has a global warming potential, or a GWP 100, as we say, of 28. So it's potentially 28 times more potent than CO2. Now, this metric for a way of measuring methane is quite controversial at the moment. And I'll talk later in the slide, in the presentation, about other potential metrics you could potentially use, because with methane, the actual persistency in the atmosphere is a lot shorter than CO2 and other greenhouse gases, N2O also. A big problem in production is that methane is responsible for a loss of feed energy in the animal. So it results in feed inefficiency. So for the animal, producing methane is a very wasteful process. So any efforts to reduce methane emissions will be of benefit to the production and the performance of the animal. As I said, we're under international and national mitigation commitments. Internationally, with the Paris Agreement, we're under international commitment to limit global warming to well below 2 degrees and pursuing efforts to really bring it under 1.5 degrees. By the EU, we're hopefully going to reduce greenhouse gas emissions by 2030 by 40% based on 1990 levels. And Ireland is required to reduce it by 30% at least. And there's also a requirement for a 2% decrease in national GHGs per year, going from 2020 to 2030. As you would have heard from the department, there's also the National Climate Action Plan that says we need to reach carbon neutrality by 2050. And you would have heard from Dale on the climatized document that the government has produced in 2019. And that really requires us by 2050 to be carbon neutral, but also to have reductions in the area of 24 to 47% when we're talking about methane. So how is methane produced? It's produced here, as you'll see in the diagram, when feed is ingested by the animal, it gets into the rumen, this large belly part of the animal here. So the plant material or the concentrated feed gets into the rumen, and there's bacteria in that rumen that digest the feed. But as during that feed fermentation process, hydrogen and CO2 is produced. The methanogens, another group of microbes in the rumen, use this hydrogen and carbon dioxide to produce another chemical or a compound called methane. And this methane is then emitted from the animal, and this is our potent greenhouse gas that we're talking about today. So it's produced in the rumen or the belly of the animal by a process called rumen methanogenesis. And this is just the formula involved, and the methane is there in red. So when I talk about CH4 for the rest of the presentation, you'll know that I'm referring to methane. So in the belly or the rumen of the animal, it contains lots of different groups of microbes. And these include bacteria, archaea, and anaerobic fungi. So the anaerobic fungi and the bacteria play a major role in the feed digestion, mainly the bacteria, as I spoke about earlier. The ciliate protozoa produce a lot of hydrogen, and then the archaea and the methanogens I spoke about, they actually use a lot of this hydrogen to produce methane. We know less about the viruses, but there is some exciting research in that area also. So fibrolytic are microbes that break down fiber in the feed, produce a lot of, produce basically the methane, that produce the hydrogen and the CO2, and then the methanogens, as I said, go on then to produce the methane. So methane emissions, if we're going to study methane emissions and try and find strategies to reduce it, or abatement strategies as I speak about, we need to be able to measure it very accurately. And there's different methods to measure methane. And we have different types of projects, be it out on pasture, where we need to measure it, or else indoors. So we have different types of measurement strategies or types of equipment to measure it. And I will say that in only recently that we've got some equipment to measure methane, we haven't had a huge amount of resources in terms of methane measurement equipment in Ireland until very recent times, and it's through a number of research projects that we got some equipment. We have the respiration chamber, which is a large box that the animal goes into that you can measure methane and oxygen and CO2. It goes into this box for a number of hours. Out in the field, we have what we call the SF6 system and tracer. So they have little canisters or like a little backpack on the back of the animal, and that they use that while they're grazing. And then you can bring it into the lab and measure the methane that's emitted. What we use more so in our projects at the moment is a green feed system. And this can measure methane usually indoors, but it can also be bought outdoors, as I'll show you from some other colleagues of mine in Moore Park are bringing it outdoors to measure methane in pasture-based systems. So this is very useful. They're using this internationally in Canada, in Europe, and basically when the animal comes in to feed, they can measure the methane. And usually it takes a long number of weeks, maybe probably between roughly 3 weeks to get accurate measurements of methane. The next thing I could talk about is reporting methane output, because you see methane being reported using different types of ways to report it. We often refer to daily methane output, which is methane, the grams of methane produced per day. The methane yield, which is the grams of methane per kilogram of dry matter intake. So we know that methane production is very dependent on feed intake. They're related. So it's important that we actually control for dry matter intake when we're doing our measurements. So that's the methane yield. Also, we talk about methane intensity. This is an important measurement because it's the grams of methane per kilogram of carcass weight. And carcass weight in beef production is what we measure. If this is dairy, it'd be milk protein is what we would use in this case. But for beef production, it's carcass weight that we measure. So this is important because it's based on output. So how are we going to reduce methane emissions from agriculture in Ireland? This is the big question. So I'm going to just talk about some of the strategies, starting off from feed efficiency or from farm efficiency, going right through to some of the exciting technologies we're developing in research projects. The first thing you need to do is have improved management practices or improved farm efficiency. And you hear talk about this all the time, and it's actually detailed in the Teagasc MAC curve or the Teagasc marginal abatement cost curve. And some of the things they talk about there, the practices include extending the length of the grazing season, increasing dairy cow genetic merit by the EBI, optimizing age at first calving, increasing the daily live weight gain of beef, cattle and lambs, optimizing the calving and lambing rate, and having then a lower age at which the animal is slaughtered. And ICBF are talking about this a lot more recently as a way, as obviously if the animal is on the ground for less time, you know that we'd have less methane emissions produced. Also, I'll talk about manure management is extremely important, as manure is another source of methane emissions. If we look at the MAC curve produced by Gary Lanigan and Trevor Donlan in 2019, looking at improved farm management, we can see that methane abatement could be estimated to be reduced by about 1.85 millitons of CO2 equivalents. That's how methane is measured in the inventories per year. And these are cost negative strategies. And not only are they cost negative strategies, they actually improve your profitability on farm. So it's a win-win situation to improve your farm efficiency. So some of the things then we're looking at moving on to the future, some of the things that we can add to these farm efficiencies include breeding initiatives. And we, Teagasc and ICBF together, are working and collaborating on trying to develop breeding strategies so you could have breed for low methane emitting animals. This goes hand in hand as well with feed efficiency. There's a direct relationship between methane emissions and feed efficiency. Animals that are more feed efficient, and that is require less feed for a certain amount of gain, these animals will also produce less methane. So again, another win-win situation. So the strategy should be really from a breeding perspective to breed animals that are more feed efficient and produce less methane. Some of the work that I'm involved in myself is in trying to develop new feed additives. So these basically are dietary feed additives to reduce methane emissions. And the important thing here is that these would be able to be cost effective, have no negative effects on meat and milk products, and can easily be delivered on farm, and also be able to be delivered on pasture, which is a critically important thing for our production systems. So that would involve developing things like a slow release option, or a slow release bolus that would be given to the animal, at certain times of the year. Another thing I'm going to talk about is multi-species swards. So there's a lot of work in Moore Park and a lot of work internationally as well. And we're going to be starting a project in Grange on this soon on using multi-species swards, sometimes in combination with additives, and to see does that reduce methane emissions. So breeding strategies. We know that the sustainability of ruminant livestock production can be enhanced by including methane emissions output in the index. I think this will be very important going forward, and ICBF are very engaged in this process. And good breeding decisions are cumulative and permanent, so that's important. And we know from the literature that breeding is an effective long-term solution for reducing methane emissions. And internationally, that would be what is expected. So what's involved? So us in Ireland, when we're trying to develop breeding strategies, what did we need to do? We need to record the measurement, which is methane measurements, on large numbers of animals. We need to collect DNA from those animals and identify genetic markers associated with methane emissions. And these could then feed into genomic selection program. We also need, it's the microbes that produce the methane in the rumen, we need to have an enhanced understanding of the role of both the rumen microbes and also the diet that the animals are on methane emissions. And we have a lot of projects working in this area. And another important thing, if breeding strategies are developed, with beef cattle indoors on concentrate feed, we also need to make sure that these breeding strategies are effective and tested on pasture-based systems. So some of the research projects we're involved in. So myself, we have a project called Rumen Predict, which is funded by the Department of Agriculture, Food and the Marines, and that's an AirNet project. Also, we have a project called Master, which is an EU Horizon 2020 project. And these are international collaborative projects, and their aim is to link the rumen microbes, the host genetics, and performance to benefit methane mitigation strategies. And we're collaborating with UCD, the ICBF, and other institutions with this effort. And these projects have allowed us to buy equipment into both Tully Performance Testing Station in County Kildare and Teagasc Grange to be able to measure methane in large numbers of animals. So this has been very effective in recent years. So the Irish Cattle Breeding Federation, as most people will know, is the organisation in charge of recording and processing all the data in Irish cattle breeding, measuring methane in large numbers of animals. It was naturally through collaboration with the Irish Cattle Breeding Federation that this had to happen, so that they're our partners in these projects. Also, my colleague Professor Jonah Berry is leading a big project called Green Breed, which is funded by DAFM. And that is also, a lot of that work is based in Tully as well, where they're developing breeding strategies for low methane emitting sheep and cattle. Through all these projects, we've been able to, I suppose, purchase and get equipment set up to measure methane. We also have portable accumulation chambers that are run by Dr. Fiona McGovern in Athenry. We're measuring methane measurements on sheep also. So there's breeding programs hopefully in pipeline as well for sheep. So in Tully, which is the Irish ICBF progeny testing station, they perform tests on over 600 beef cattle per year of various breeds and various sires. They measure feed intake, feed conversion efficiency, average daily gain, and they're linked to projects to measure meat quality also. And the cattle undergo a 120-day finishing period. So this serves as a really ideal opportunity to build in another measurement on top of these measurements, where we could actually have methane measurements. So we could actually do some correlations with these other traits, and we can look at the effect of methane measurements on those other performance traits. And also ideally, we can actually also collect DNA samples and work on developing genomic selection breeding programs. There's a 30-day acclimatization period. There's a 90-day feed efficiency period where feed efficiency is measured, which is very useful. And then we do a 21-day methane measurement period using the green feed systems. The steers and heifers, they feed on hay, concentrate and hay diets. We have green feeds installed. We have at least 6 installed now, at least, and not if not more coming and people being trained up on them. So this is a great initiative. And then we'd have samples collected where we can actually collect rumen samples from the animal, and these are analysed to see what microbes are in there, in what abundance in the rumen of the animals, and that's done in Teagasc. So just looking at some of the research to date and the output from this research, already we have looked at 400 beef cattle have been sampled, and this is through a project that one of my students has, Paul Smith, who's doing very good work there, and Stuart Kerwin. They go to Tully and do a lot of this work. Also, the cattle are producing an average of 224 grams of methane per day. Some of the preliminary data from these animals, we've just done some analysis, and the preliminary data shows that enteric methane emissions and feed efficiency are negatively correlated. So what does that mean? That means that low methane producing cattle are more feed efficient, so require less feed for gain. So you have feed cost savings as well. Um, and so that's a very exciting result. So we ranked all these animals and we looked at two cohorts, the two extremes of these animals, and we find that low CH4, low methane emitting animals produced about 30% less methane and 30% as well less methane intensity relative to the high methane emitting animals. And feed and actually performance was not compromised. So basically, the animals were still growing at the same rate. So this is very exciting work, and this has been written up into an abstract or a summary to be presented at this year's British Society for Animal Science by Paul Smith. So have a look out for that. And then looking at the breeding index. So a lot of people listening may be involved in the BDGP and will be familiar with the terminal beef breeding index. So really looking at the relationship between daily methane and the index, we find that animals that have a lower methane yield have a higher ranking on the index. So that means that animals that are in your 5 stars, 4 stars, 5 stars, these are actually producing less methane. And reducing methane emissions then will allow us to enhance profitability. The next thing I'm going to talk about is multi-species swards. And so this is another area of research that could be useful for us for reducing methane emissions. So we know in Ireland, of all places, that grassland pasture grazing is the most sustainable form of livestock production. And when cattle are fed a high legume proportion diet, research has found that there's 20%, up to 20% reductions in methane emissions were observed. We look at white clover. We can see from work at Moore Park that this increases passage rate through the rumen and could also potentially impact methane emissions intensity because looking at feeding the white clover, they got some increased milk yield, milk solids. So this in turn allows us to have lower methane intensity. So for the same methane production, you're having higher levels of actual milk production. So that in turn will reduce methane intensity. So it's a win-win situation. And then looking at some pasture herbs, they find that some of the bioactive compounds that are in there include condensed tannins. And these could be this could be the compound that is causing the reductions in methane emissions. And there's a lot of research on that at the moment. Even looking at mixtures of sorrel and oxeye daisy and other multi-species swards, when these are fed as haylage, that you've seen 10% reductions in methane emissions compared to feeding perennial ryegrass or the normal grass monoculture. So this results in improved animal performance, if you to summarize it, with livestock grazing multi-species swards, and also results, as we see from the research, in having lower methane emissions or reduced methane intensity. So that's a win-win situation. But there's also another co-benefit when you're talking about multi-species swards, that there is a potential for dual greenhouse gas abatement, as legume inclusion with swards can reduce nitrogen fertilizer requirement. So it reduces N2O emissions and overall emission intensity of the grassland production. So we are working in this area at the moment in Teagasc and Moore Park and Grange and Moore Park. So this would be potentially another avenue that we could be pursuing in the future. In work that we've done ourselves in collaboration with Moore Park, we looked at the effect of white clover on the abundance of rumen microbes, so the microbes that exist in the rumen of the animal. So working with Deirdre Hennessy and colleagues in Moore Park, they found that white clover inclusion resulted in 12% reduction in methane yield, while there was no effect on milk yield. We then looked at some of the microbes, or particularly those methanogens I spoke about earlier, and the abundance of those methanogens in the lab in the Animal Bioscience Department. And we found that there was a decrease in the relative abundance of Methane brevibacter in the white clover compared to the perennial ryegrass. And this was a very kind of exciting finding in that, it's looking like clover is affecting the methanogens in the rumen of the animal, and this could be a mechanism of action. And we published these findings with other findings more recently in a journal, the Scientific Reports, which is a Nature paper. Now, just moving on to feed additives, and one of the projects that I'm working on myself is called Methane. It's funded by the Department of Agriculture, Food and the Marines, And it's on the development and validation of novel technologies to reduce methane emissions from pasture-based Irish agricultural systems. So this is really trying to develop effective feed additives that can be used on pasture-based systems in Ireland. So feed additives, we're trying to find feed additives to mitigate methane emissions, and we're looking to monitor their effects on animal productivity at the same time. So ensuring that there's no negative effects on animal production. So what kind of feed additives are we looking at? A lot of people would have heard of 3-NOP or Bovar by DSM. Seaweeds, and we also have another project I'll talk about in a minute, which is Sea Solutions, that's working on seaweeds and seaweed extracts, oils, halides, yucca extracts, and olive feed preparations as well. We also have some tasks within this project that we can try and develop encapsulation, and this is done with NUI Galway, our partners on the project. They're trying to develop slow release options or slow release boluses that can be delivered at pasture. We're also focusing on the nutritional and toxicological composition of meat and milk to confirm consumer safety, that any additives will have no residues or negative effects on meat or milk production. And of course, to ensure sustainability of the system, that that there's no issues with sustainability in things like seaweeds or the production of these additives, we're going to carry out Teagasc life cycle analysis. And then an important thing would be the farm level cost effectiveness is extremely important. There's no point having a feed additive that works very well if it's too expensive to deliver to farmers. And this will be carried out in collaboration with the National Farm Survey. We're in the process now of testing a lot of these feed additives in the laboratory. So we have a system called the Russitech system, which contains rumen fluid, where we're testing these in vitro before going to the more expensive animal trials. And that's ongoing in Teagasc Grange at the moment with my colleague Stuart Kerwin is running that part of the project. The next step then is basically fine-tuning which are the most effective feed additives in vitro, or as we say it, or in the lab, and then bringing those to the animal trials. We have another number of animal trials planned in Athenry on sheep, in Grange on beef cattle, and also in Moore Park on dairy cattle in 2022. As I mentioned, one of my colleagues, Dr. Maria Hayes, she is actually leading a project called Sea Solutions, which we're involved in. And this is looking at seaweeds and seaweed ingredients to reduce enteric methane emissions from pasture-based sheep, cattle, and dairy cows. And this is has a multi-partner European project. So we're working with many partners on this. And to date, we have 11 seaweeds screened in vitro for anti-methanogenic properties. We found no effects on the brown seaweeds, which we see a lot of in Ireland, which would be a good option. We only see reductions in Asparagopsis taxiformis seaweed, which is largely highly reported in the literature anyway as being effective. But this would be a very expensive option for us in Ireland. And future work will allow us to evaluate different seaweed extracts like the tannins we talked about earlier and peptides in the system. I'll just mention in Moore Park, they also have the outdoor green feed system as part of Vistamilk, and they're evaluating feed additives and pasture-based systems. And that's Dr. Lawrence Chalou in Moore Park is carrying out those studies, working with Katie Strasmore, and that's part of the Vistamilk project. Next, I'm going to talk about, you hear a lot in the media and elsewhere, is on the metrics for measuring methane. So, as I mentioned earlier, the IPCC measures methane using global warming potential of 100. And this is the current metric used to assess the impact of methane on climate change. This equates to 1 GHG units to its CO2 equivalent averaged over a period of 100 years time horizon. And this gives methane a GWP of 28, according to the IPCC, which carries out our inventory, which basically calculates these metrics and tells us what metrics we should be using. We find, though, however, and this is reported in a lot of the literature in some of the papers I mentioned here, Allen from Oxford and Lynch is part of that team as well, and they're basically saying that it fails to account for the short-lived atmospheric persistence of methane, which is roughly about 9 years relative to other GHGs. And this is important to consider because you see N2O and CO2 lasting, 100 years anyway in the atmosphere. So what we're saying is, does this overestimate the contribution of methane to long-term radiative forcing or temperature change, which is ultimately the measure that we're looking at in terms of the IPCC? So there's a new metric that people are talking about. It's called GWP star. And this is a useful indication of warming and means of assessing alternative mitigation strategies. So GWP star is an alternative metric that's potentially going to be assessed here, that should be used, but that we cannot just simply start using it. It will have to be assessed by the IPCC, and it will have to be agreed by the UNFCCC. So currently, the FAO, as part of LEAP, are going to assess this metric as a measure of methane emissions. It will be compared to GWP 100. But still, even with this, we still have to reduce methane emissions and develop mitigation strategies. So this is a useful paper if anyone's interested in this topic. It's by Lynch et al. It's about GWP star. As another role as part of my research, I'm also wearing another hat. It's the co-chair of the Livestock Research Group of the Global Research Alliance for Climate Change, which basically is funded by the New Zealand government. And now we have another co-chair from the UK as well. And really, it's part of this Global Research Alliance for Climate Change. It covers like 65 countries, and the agenda really is to grow more food without increasing greenhouse gas emissions. And I work with my colleague in the Department of Agriculture, John Harrison. He is the Irish rep as well, so that we can actually be involved in some of these discussions. The GRA overall plays a big role in research and trying to bring the research to application through networks. And we have a number of networks that we coordinate. We have networks on manure management, on rumen microbiome, on feed and feed network, genetics network, and animal health network. And there's lots of scientists working within those networks, trying to come up with solutions, working together, writing up projects together, and it's very, very effective. And I would recommend to any researchers out there, our industry partners, to get involved in the GRA. But this role allows Ireland to have a role in discussions in GHG mitigation, tier progression, and the national GHG inventories. And I suppose it's also got us involved in the United Nations FAO, where the discussions on there, we write reviews and get involved as well with LEAP, the Livestock Environmental Assessment and Performance Partnership, which is likely where the GWP star will be assessed. So this is the GRA. So just to summarize, just don't want to go over time. And to summarize today, I'll say that methane is a potent agricultural greenhouse gas. We're under national and international commitments to significantly reduce it. The first thing to do would be to enhance farm efficiency. But we also have, on top of that, some promising research currently ongoing in Teagasc, in elsewhere and around the world, on developing mitigation strategies, breeding strategies, feed additives, and potentially the inclusion of multi-species swards, and potentially using these together. Also, things to be aware of is the methane metrics, and I do believe there will be some work on assessing some alternative metrics in the future. But in saying that, we still need to develop mitigation strategies and to reduce methane significantly. And I think it's important for us to stay in touch with, and have international collaboration. It's important for Ireland, and particularly to link in, with other grass-based countries like New Zealand. And we're going to have a speaker from New Zealand next week who's going to tell us some of the things that are going on in New Zealand, Sinead Waters. But she's also involved in the GRA. So that's it.
Thank you, Sinead. That was really excellent run through of the enormous work that's going on in this area, and congratulations to you and the team, your team, for the amount of work and output that you've achieved over recent years. A huge amount of work going into this area of feed additives. Just Sinead, if you could maybe stop sharing your screen there. My question was in relation to the whole area of feed additives, and how confident are you that this area of work is going to actually deliver reductions in methanes over the next 10 years?
Yeah, so in some of the projects that we have ongoing now, we're seeing some very good, promising candidates coming through from the lab, and that will have to be tested, of course, in animals. If we look at the literature, we can see from one of those feed additives, for example, the 3-NOP from DSM. This is a feed additive that has shown consistently around the world that you can reduce methane emissions by, at the minimum, 30%. It can sometimes, if you feed a higher level, can go up to 41% or more. And I think they're trying to develop now as well. They're working with us in Methane, but they're trying to develop a slow release option that could be delivered at pasture. And we will be testing that within our project. But if that was the only thing I would see is to get it sufficiently cheap, so that it can be delivered on farm. That would be, I definitely think it will work because it has worked in other jurisdictions. I think that so with the slow release option, I think that's the way they will get it sufficiently cheap to be delivered for us here. There are other options coming through as well that are looking promising in the lab that are reducing methane emissions quite significantly. And these will be tested, as I say, in sheep and in cattle. And we're also going to look at maybe using 2 additives or 3 additives together, if that was potentially an option, to see if they had any additive effects. And we're also going to do some experiments where we're going to feed the feed additives on multi-species swards to see if there's additive effects there. So yeah, I think with a slow release option, and that could be delivered in the dairy sector on pasture, that would be a very good option for us. I would be confident by maybe 2025 that we would have some solutions, hopefully.
That sounds very exciting. I know there's a lot of concern about, the use of seaweed and the ecological impact of harvesting seaweed. But I think the longer term goal is to create a synthetic version of those. Is that right?
Yeah, so we hear an awful lot about seaweed in the media and elsewhere. And some of the seaweeds, for example, Asparagopsis taxiformis I spoke about, they're the red seaweed that's kind of used. It's been tested in Australia and Canada. That can reduce methane emissions quite significantly, like up to 70%. You can see in some studies, and absolutely, that's grand. But the red seaweed for us, if we were to purchase that here, would be just we couldn't afford it. It's not an option. The other side of it is that some of these seaweeds contain compounds like bromoform that are potentially carcinogenic to if the residues got into the meat or milk, that you could have bromoform, and that is a carcinogen essentially, so that would have to be tested for human safety. They say internationally some studies that they don't detect it, but we would absolutely have to, here in the EU, definitely have to test that for residues in the milk and meat products. But I think absolutely sustainability is an issue. Can we produce enough seaweed in Ireland to feed all the animals? I just think I think the way forward, and it's through some of the work that my colleague Dr. Maria Hayes is doing in Sea Solutions. We're working with her on that. Is trying to she's doing some very nice work on characterizing the seaweeds and looking at the components of those seaweeds and extracts. And we're going to test those now actually in the coming weeks in the laboratory for their antimethanogenic ability. So if we hit on an extract that's very effective at reducing methane, we can potentially produce that synthetically, and that could potentially be an option for reducing methane emissions. I think that's the best route to take.
A question here in relation to, I suppose, the efficiency element. I mean, one of our viewers here agrees with all of the points, but surely it has to be in the context of stable herd is the question here.
Yeah, that's a good question. And that always comes up. We see, we should just reduce the herd. I think that, I wouldn't recommend anything drastic in terms of reducing the herd. And I can appreciate it depends on, the herd size, the total methane emissions. But I think that, based, even if it is a stable size, the herd, we do need mitigation strategies. And if we can reduce sufficiently, that can't be compromised,
Okay, the questions are coming thick and fast, Sinead. So I think we'll probably try to have condensed responses if possible.
No problem.
And I should have introduced our colleague Pat Murphy earlier on. Pat is our head of knowledge transfer on the environment program and based down in Wexford. And you're going to help us out with questions, Pat. A lot of questions coming, huge degree of interest here in this topic, Pat.
Yep. A huge number of diverse questions. Do some breeds of animals produce more methane than others, or is it more an interbreed or an intrabreed variation?
Yeah, so in Tully, this would be a good opportunity to study that, Pat, because we get lots of breeds coming through there. Just looking at our own data from my student there, Paul Smith, is doing a lot of work looking at the different breeds. And we're seeing just from preliminary data is that like the Limousin looks to produce slightly less methane than some of the other breeds, Charlies and others. But as I say, we'll have to do a full statistical analysis on that. But it looks like there's some breeds are in fact producing less methane.
Okay, and there's a question on the difference between high concentrate and high grass diets, and noting that you've a lot of the research has been done on high concentrate, but
yes.
what's the findings in relation to that, or what's it telling us?
Yeah, so generally we find, say, less methane produced on forage-based diets, and we have a very efficient, say, grass-based system in Ireland. So on grass, they should produce less methane. But what I said there earlier, and it's very true, is if we find, something that's successful, it has to be validated in pasture-based systems. And because you will get re-ranking. If you look even at something like feed efficiency, and you measure animals for feed efficiency and you put them out on pasture, you will get some level of re-ranking. So the same is true for methane, because I suppose it's directly linked to intake as well. So we will, and that's a very good question. So be it if we develop breeding strategies, these will have to be tested in pasture. If we have an additive, and that's why we are going the route we're going, testing in the lab, going to sheep and beef, and then testing a pasture with dairy as well. Actually validated across the species. So it is a good point.
A question there about the quality of the fodder that we're feeding to our animals. Are you seeing differences between low quality silages and hays versus high quality feed?
Yeah, we haven't assessed that, but we will be when we actually get to the beef trials, because we're going to do some new different types of diets, and that's the point that I made earlier, that the diet is a very, very important factor in this, because I guess it's directly linked to fermentation or digestion in the rumen, and that's how methane is produced. So I would imagine that you will see differences on the diets.
An interesting question. You say most of your research is on finishing animals. What about the suckler cow itself? And I suppose there's an issue there where you're less likely to be feeding a supplemental feed to those dairy cows. Is there anything we can do there?
So yeah, that's a good question. In some future studies, we are going to actually be feeding younger animals and the suckler cow. That's part of some of the work we're going to be doing actually later on. But there will be ways to get the supplement into feed. Just even like some of the supplements are just like a quarter of a teaspoon of the additive. And you can get it in, even if it's younger animals, to even potentially dose them, in some way early in life. But there will be a mechanism to get it into the animal, absolutely, because we're working with industry, and they'll definitely want to get their product developed for all types of animals.
There's a question here about what do you mean by slow release, and why is it necessary?
Okay, that's a good question. So when animals are out at pasture, you might have an opportunity to bring them in to give them the additive. So what they want is basically an option that you have the feed additive in the rumen of the animal. So it's encapsulated, that it's released gradually over time. So there's encapsulation processes that you can do to do that, like into a bolus. So the animal is given the bolus maybe once every 3 months, and slowly released over that period of time, so that it can be effective while they're out at grass.
A question there: at what percentage of clover did you start to see some positive impacts on the diet? On the emissions.
Yeah, I'd have to look back at that period, but it's roughly, when I think from like about 20-25%, around that kind of thing. That's some of the work at Moore Park. We didn't really look at percentage. It was actually like a mixed perennial ryegrass and clover, and then compared to perennial ryegrass, but around from about 25% onwards, I believe.
Okay, a question there in relation to, sorry, I'm lost it here. Oh yeah, in terms of you mentioned life cycle assessment in relation to seaweed. What do you mean in relation to that?
So, if you look at seaweed production and seaweed, if we're producing lots of seaweed also produces other greenhouse gases such as ozone. So you want to make sure that in the production of your feed additive, that you're not generating more greenhouse gases. That's just an example for seaweed. So there might be some environmental cost to producing some of these seaweeds or some of these additives, and you don't want that. You don't want to have added. So that's why a full life cycle assessment will have to be done.
An interesting question here: the potential impact of the products that we're looking at. we're looking at it very much from a grass-based system. Other countries are looking at it more from obviously the indoor feeding system. Which system do these technologies provide most advantage to, and will it change the relative greenhouse gas emissions of the two systems?
Yeah, so there's only more recent work in, the person is right. A lot of the work has been done, say, indoors on finishing animals or, on high concentrate diets. So there's been less work done on pasture-based systems, and that's why this project Methabate is going to analyze that, as well as Lawrence's project in Vista Milk. He's looking at say, dairy cows outdoors on grass, and I think we'll have a lot more answers after that. And you're right, that's more important for us. For New Zealand, they're also looking at grass-based systems, so they're looking at feed additives outdoors. And I would agree, a lot of the previous research was done indoors, so on concentrate diets. So we'll have some data, coming up in the next couple of years on that.
A couple of questions in relation to the long-term impact and I suppose the animal welfare impact of these products fed on a long-term basis. Any initial findings or initial observations in relation to it?
Well, what I'd say is that from the studies done internationally is that that's very important, and we will be monitoring, of course, for animal health and welfare. That's critically important, like we're monitoring for performance. There hasn't been anything really shown internationally on some of the additives that were tested, but these additives would have to go through a process beforehand to show they were safe. So, that would be critically important. Even for us here, we'd have to go through the Department of Agriculture, make sure and through our ethics committee, and then through HIPRA. So we actually would ensure that that's critically important, and that would be assessed before we even start a trial. But we would absolutely be monitoring. We would be monitoring for those.
There's two other additives we mentioned here, Sinead, biochar, charcoal. I presume they're close relations. Is there any data showing the effects of these in terms of methane production?
Yeah, I know there's some trials going on, and but I haven't seen very much data on that yet, Mark. We're not assessing it within our project, but if there was some opportunity maybe from industry, it could be facilitated. But I haven't seen very much data from the biochar side of things, but I do know the research is ongoing.
In relation to the GW Star, a lot of questions and interest in terms of the timeline or potential timeline there for this coming into actual policy. I suppose that's a very difficult question to answer.
Yeah, no, I actually raised this issue as part of my work on the GRA at our annual meeting, and I said, for countries like Ireland, it could be important. The issue here is as of course everyone knows, is the short lifetime of methane in the atmosphere compared to CO2 and N2O, and that needs to be addressed. We will not be able to change that here ourselves. It'll have to be done through the IPCC. So really what's going to happen is, which is great, as we have actually contact with the LEAP, which is part of the FAO in Rome, and they're going to assess whether this is an option. So they do a lot of modelling, and they carry out, they're going to do some direct comparisons between GWP 100 and GWP Star. So there were some very interesting presentations on that in September, and our own Minister for Agriculture actually gave a short talk at one of those presentations, and that will be actually addressed in the future under that banner. And I think data from that then will feed into the IPCC.
I suppose it doesn't get away from the fact, though, that we still have to reduce methane. Absolutely. I think it's not a get out of jail card.
It's not, and I think we can't be naive here either, because I think it doesn't matter. It'll still be a way that we're going to measure and look at mitigation strategies under GWP Star. So if we get it, if that comes through, or maybe a variant of it, a type of GWP Star, we'll still have to reduce methane emissions. You can't get around it, but we are potentially being penalised heavily, as well. So we just need to have a balanced view of it.
You mentioned methane intensity in your presentation, Sinead, and it probably deserves a whole talk on its own. But I mean, this is a really crucial point, isn't it, in the whole greenhouse gases debate? Particularly when it comes to vegan diets versus meat or milk or animal diets. Could you
Yeah, so
care to
methane
comment on that?
intensity, yeah, so methane intensity as a way of measuring methane is based on the product you're producing at the end of the day. So like there's no point really in measure. It's a very important measurement of methane because we should be totally focused on, for milk protein and beef per kilogram of beef. So really there are our metrics. So it's important that's our output from agriculture. So we need to be, I suppose, cognizant of the fact of how much methane we're producing per output. So that's why it is an important, and I think people are expressing methane emissions in that format, in research and in conferences because of that importance. Like,
just a reminder to everybody that today's presentation and recording of the webinar will be available on the Teagasc webinar page. So just a few questions coming in on that. Pat, a few more questions there, I think.
Yeah, I suppose a couple of questions in relation to multi-species sward and the mechanism of that you see that is having the impact. Have you any idea what it is?
Yeah, I think, Pat, really that it's potentially some of the tannins and the bioactives in, say, some of these like clovers or herbs. So that's similar to the seaweeds. That's something that could be assessed. But I think with multi-species swards, we can get many benefits, as I said there in the presentation, because you can get an increase in production. And also it limits the amount of fertilizer that we need to use. So there's lots of co-benefits with the multi-species swards, I think.
I suppose a lot of questions about the size of the national herd and
of course,
whether there's enough in the long-term reduction through methane to offset to the levels that we need to achieve the type of objectives that are there. So I suppose what a lot of the questions are alluding to is, where do you see the potential, or to what level do you see the potential, either under the current measurement system or the GDPR star, to try and get us to where we need to be? What kind of hope do you have for the capacity of methane to contribute to reductions from bovine animals, ruminant animals, I suppose?
Yeah, so I guess, we'd be all very reluctant to start, reducing the national herd. I think we need to look into some of the strategies that we have. And as I said, okay, farm efficiency, that's the first thing. Second thing is with breeding, we see already in the data coming from Tully that there's a difference between the highs and the lows of about 30% in methane emissions. And that's both methane output and intensity. So that's very promising that you could potentially have a breeding strategy. On top of that, you have the feed additives. We could potentially have a 30% reduction if we have 3-NOP, and that works as well for Irish production systems. And then there's an added benefit if you have those animals that are of, say, the 5-star, the high EBI, or that are bred for low methane emissions. If you have those animals on with an additive and on a multi-species sward diet for some of the year, some of the time, that potentially we could reach what we are looking for in this. So if you combine all the strategies together, and we're at probably a very important juncture in that, in the next couple of years, some of the research will be coming out of these projects, be it our own projects, our Vistamil, Greenbreed, the work from Tully. And I guess a combination of those is what's, going to work. But some feed additives are very promising compared to others. And I suppose it's getting into the stage that they're validated in the beef and dairy cattle is what we need to get to. But I would be optimistic.
Okay, a more straightforward question. There's a number of people have asked about the impact of earlier calving and earlier slaughter. How does that have an impact?
Well, that has an impact because the animal is on the ground for less time producing methane, I guess. So there's less days alive, say, for producing similar output of beef. That means that it would produce less methane.
A question in relation to the I suppose the correspondence between the in vitro studies and in vivo studies. Are you getting a high level of correspondence when you're progressing from one to the other?
Yeah, we haven't carried out in metadebate now. We haven't gone to the animal trial work yet, okay. So we can only go on, say, international studies. They won't be exactly the same, but it is the ROSI check system, the system that we have in place that mimics the rumen, is, it's a very good system in terms of it's rumen fluid from cattle or that's in there, and it just mimics it. So there should be some correspondence anyway.
Any work on some of the older breeds, or is there anything from the breed side which indicates bringing back other breeds that might have disappeared over the last number of years?
Well,
like
we haven't.
Shorthorns or.
Yeah, look, we haven't looked at those, but we could potentially in the future. We're seeing some differences in breeds. We'd like to have more data before we'd say anything about that. But yeah, potentially we haven't looked at those breeds, so I can't comment, I suppose, on it.
I think we've covered a lot of the questions coming through today have,
very efficient,
similar themes. And, I suppose, I mean, that question that we keep coming back to during this entire series is the cow numbers or animal numbers in the country. And I guess if we're, honest, I mean, obviously there has to be, at some point, there has to be some form of the expansion can't continue forever. I mean, a lot of the research and the efficiencies will bring us so far.
Sure.
So I think it will be disingenuous of us to say that.
Oh, yeah.
That
I think as well, like, we can just generate the research and the results, and I think it'll be up to policymakers to decide on that. I don't think that's really our remit in this.
Absolutely. I couldn't agree more. And I think that's really, I mean, and this is why maybe some people think we're being sidestepping the issue in some degree, but in fact it is, quite like you say, it is a decision for policymakers to make. And so, but I mean, there's a simple arithmetic that goes on there. Sinead, I think we've covered most of the areas. I mean that's through questions and your presentation there. But in terms of like casting your eye 10 years down the line, what do we see as the animal of the future? In your mind, with all of the different work that's going on in genetics and on the environment that those animals are living in.
Yeah, I think that even if you look at dairy production, like, they've come an awful long way with the breeding. So, and I think we can similarly across beef and dairy potentially be breeding for low methane emissions with the research that's going on. And I suppose the really good setups that are in working collaboratively with the ICBF on that, and the methane measurement equipment that we've in place. And I think we're really improving things that way. So if you had an animal that is bred for low methane emissions and high feed efficiency, because that goes hand in hand with it, because that will reduce feed costs, which is very, very important, and enhance profitability. So that's the kind of animal you'd have of the future. And if we need then added feed additives will become, I think, less costly and integrated into the feed, be it for during concentrate feeding or as a slow release bolus for when animals are out of pasture. That's the way I would see it. And that multi-species swards then could be part of the system, for a certain time of the year, when animals are out. So because that would be a way to maximize the additive effects. And of course there has to be continued farm efficiency. I see it myself, it particularly, I say, in small, suckler beef farms, to become more really, I suppose, efficient in management practices, and they would need help with that, I think, and so I think it's efficiency combined with breeding, combined with potential feed additive, and the correct diets as well, what I mean, that they'll be on, and in some cases, early age to slaughter, all these kind of things.
Okay, brilliant. Okay, Sinead, we're right out of time. Thank you so much for your time today and the effort you put into the presentation. I know you have broken down some complex issues there for us.
Okay,
so
hopefully.
We do appreciate that. So just before we wrap up, I just want to share details of a conference, or a virtual conference that Teagasc is hosting in collaboration with the Scottish Royal College. This is taking place on the 9th of February. It takes place via Zoom, and you can get full details on the Teagasc website. And the topic that's being dealt with is the whole role of digital in supporting the green or natural sustainable recovery of the economy. So we do encourage you to sign up for that if you have time. With that, I just want to thank our production team, Andy Boland, Yvonne Maher, Catherine Keena, Pat Murphy, and thank you for viewing our series today. We've huge following on the series, so we really welcome your support. And until next week, we're going to be talking about the New Zealand situation, and we'll be having perspectives from our colleagues who are in a similar climate to ourselves and similar issues, facing similar issues. So it'll be great to hear that experience. So do join us next Friday at 9:30 for the Signpost series. So with that, thanks again, and we will talk to you next week. Bye-bye.